Hello. Thanks again to Fear Free and Purina Pro Plan Veterinary Supplements for making this presentation possible. So the recognition of microbiomes as mediators of host health has been arguably one of the most profound paradigm shifts in the life sciences in the last few decades. But I should emphasize that I am a veterinary behaviorist with a strong interest in the subject, but I'm not a gastroenterologist or a bacteriologist. So this is going to be a high-level overview of the microbiome and its basic functions. And we'll also touch on the correlation between the gut microbiome and local gut health. But even more fascinating, we'll also talk a little bit about the links between gut health and distant systems. And I'll go a little bit more into detail with the gut-brain axis since I am a behaviorist. So let's start by defining some basic terms. The microbiota is the organisms. So this is the bacteria, the viruses, the fungi, the protozoa living in the body of interest. So in other words, who is there? And bacteria make up most of the microbiome. So the microbiome, though, refers generally to the genetic material and the function of the microbiota in that area. So in other words, what are those microbiota doing? And related to that is a term called the metabolome. So the metabolites are the products of the microbiota. So this also relates to the function of the microbiome. So according to some estimates, there are more than 10 times more bacteria than host cells associated with an individual, although this number has been debated. So I put that, I also provided that scale that some researchers think that, it probably is more, but maybe not 10 times more. And this is just a visual graphic of the microbiome diversity by body region in the dog. Although there's some fascinating research on other systems, such as the oral microbiome and the respiratory microbiome, the gut houses most of the host organisms' microbiota. And we have proven system correlates with the gut microbiome. So most of the research is focused on that, and that's what we'll focus on in this talk as well. So within an individual, the bacterial species are going to vary greatly depending on what portion of the gastrointestinal tract you're referring to. And it really reflects that underlying physiology of that anatomic location. So in the stomach and the small intestines, this is where much of the digestion and absorption of the nutrients is occurring. While moving further down into the large intestine, that is where microbial digestion of non-digestible material occurs. So in other words, plant material for the most part. It's also the location where most of the body's water absorption, mucus secretion, and fecal storage occurs. So the different bacteria are going to aid in those different physiologic functions. So it's important to note that most of the research, regardless of what species we're talking about, these are going to be assays of the fecal microbiome. This is the least invasive method of assaying the microbiome species of that individual, but it should be interpreted with caution because it represents only only the species that pass through into the feces. So this is going to be more of a confounding variable in people than companion animals because of the shorter GI tract in cats and dogs. So again, I am not a bacteriologist. I'm happy to get you some articles or webinars if you are more interested in learning about the different bacterial species. But in general, actinobacteria and bacteroides, they are considered to have most of the beneficial effects. So in other words, their metabolites are the most beneficial to the host, whereas Firmicutes and proteobacteria, they tend to be higher concentration in disease states. And this, again, has been particularly true in people where it's been the most examined. But it is important to know this is a huge generalization and the picture is more complex. For example, some species of bacteria in the same family can have very different functions. So Clostridium difficile, which is a firmicute, is associated with GI disease in humans. But Clostridium hyrenosis, the actions of that bacteria are critical for gut health in all of the species shown here. So people, cats, and dogs. And it's very important to look at the species itself. So firmicutes and proteobacteria are more prevalent in high protein and high fat diets. So this, of course, is going to mean that those are higher in cats. So this might be a marker of disease in humans, whereas it's a normal adaptation in felines who are obligate carnivores, compared to cats, dogs and people who are omnivores. So when we look at the microbiome and how it functions, first we need to look at this from an evolutionary perspective. You know, what the microbiomes are, the microbiota is there because it benefits the host, and the host likewise benefits from, or the microbiomes stay there because they are benefited by that environment that the host provides. So as a host has evolved mechanisms that allow microbiomes to thrive, it's because they have reciprocal health benefits. So mutualism and symbiosis, they're a long-standing story in nature and evolution, and there's no doubt that certain microbiomes and animals have co-evolved. So sort of the one big obvious example that most people give is this sort of cycle between the gut microbiome that create these metabolites, which nourish colonocytes in particular, and in return, the enterocytes can create mucus that nourish the bacteria. So if we just sort of stay with focusing on the functions of the microbiome for the host right now, you know, the microbiome is so metabolically active, it's often referred to as its own metabolic organ. And so some researchers go so far to state that the microbiome, that most mammalian metabolic functions involve the microbiome. So we just discussed one of the most well-known functions of fermentation, or include fermentation of non-digestible material. So humans and cats and dogs, we cannot digest plant material directly. We rely on microbes for that. So most of us remember from vet school, or at least you're kind of vaguely familiar with, that ruminants like cows, goats, but even horses, they are herbivores, so they get their nutrients from plants. And so it is the microbes in their gut that create these energy sources, usually in the form of short chain fatty acids like butyrate and acetate and propionate, manufactured by these gut microbes in the colon. But even carnivores make use of these pathways to fuel intestinal cells like colonocytes. But that's not all. Microbes are also needed to produce some very important vitamins like vitamin K and the B complex vitamins. And then also bile acid biotransformation is done by certain microbes. And bile acids are essential in our digestion of nutrients. So how is the microbiome protective? It promotes a healthy gut barrier. So those the beneficial bacteria that create certain beneficial metabolites, what this can do is prevent leaky gut. So prevent the translocation of pathogens and toxins and antigens from the gut lumen into the systemic circulation. And how does it do this? It can actually reduce pathogenic bacteria. It can actually crowd them out, but also some bacteria, some beneficial biota create pathogenic toxins. So they actually create antibacterial products that are detrimental to the pathogenic bacteria. And they also can modulate that local immune system so that the body can help fight off those pathogenic bacteria. So that also leads us to talking about how balanced the immune system needs to be. So the microbiome need to be recognized as beneficial, so and left alone, right, and not destroyed by our immune system. So how does that happen? The microbiome is just critical in shaping, or what we call educating, the host immune system during development. So early development, this needs to happen. So since we've already established that they're vital to the health of the host must have mechanisms to distinguish these friends from foes. And inflammation is a primary host defense against an invading organism. So the enteric or gut immune system is trained to recognize and not trigger pro-inflammatory cytokines when detecting beneficial microbes. The role of this education in the immune system early in development and the link with chronic disease has been a really active area of research. So diseases like asthma and irritable bowel syndrome are thought to be dysregulations of the immune system that may have developed due to some sort of dysregulation early in life. In addition, some micrometabolites like these short chain fatty acids that have anti-inflammatory effects, while others can tell the immune system to downregulate in the face of these friendly microbes. So what does a healthy microbiome look like? Some hallmarks are diverse and abundant microbiota, with the beneficial bacteria being the dominant species there. Production of these anti-inflammatory metabolites, such as the short-chain fatty acids, a healthy mucosal barrier, and little to no inflammatory indicators. So when the gut environment changes for any myriad of reasons, we can have dysbiosis or changes in the microbiome that impact function in a negative way. So hallmarks of a dysbiotic gut are low microbiota diversity, with the prevalent species being those pathogenic species. Dysregulation of the short chain fatty acid production, disruption of the mucosal barrier, inflammation, markers, and including oxidative stress and oxidative damage to the local tissues. So what can influence, what do I mean by changes in the environment that can affect the microbiota and the microbiome? Well, pretty much everything. So everything from were you delivered by C-section or vaginally? Of course, genetics, your stress system, exercise, antibiotic usage, or other xenobiotics, your age, your diet, things like that. So the impact of these factors on the microbiome has been documented in humans, and we're just now kind of building that body of evidence in companion animals as well. So this is a very busy graphic, but it illustrates that many system dysfunctions have been linked to gut microbiome dysbiosis. So it's important, though, to state at this time that most of these studies can only provide correlation. We got to be careful about causation. So one obvious mediator that ties all these systems together is chronic inflammation. So we've already mentioned that the anti-inflammatory effects of some microbial metabolites, most notably the short-chain fatty acids, and these appear to not only work locally in the intestines, but can also diffuse and act systemically. Likewise, inflammatory cytokines are also thought to diffuse and theoretically cause some of these negative systemic effects. So what diseases are we talking about that have been linked to dysbiosis? So of course, let's first start locally at the gut. So one gastrointestinal disease that's received much attention in both people and pets is inflammatory bowel disease. So in people, diseases like Crohn's and ulcerative colitis are types of IBD. In veterinary medicine, we more commonly refer to this as chronic enteropathy now. And in our pets, you can see diarrhea and vomiting, weight loss. In cats, constipation is definitely a common clinical sign with chronic enteropathy. And these diseases have been shown there can be genetic and environmental factors. And these environmental factors that shape the microbiome and immune system early in development, again, may be the most impactful. So an IBD in most species studies is associated with dysbiosis and widespread changes in gut physiology. So a decrease in the beneficial microbes results in decreased anti-inflammatory factors and other metabolites that affect gut homeostasis and immune tolerance. So the conditions can become more favorable for pathogenic microbiota and dysbiosis. And this is sort of a cyclical effect, and the disease, these two get worse in tandem, that the disease signs and the dysbiosis can get worse as the disease progresses. So if we look at other systems, such as gut health and the brain, this is a system, the gut-brain axis has received extensive attention in the last decade or so. And so what is the gut-brain axis? It is this complex bidirectional communication between the gut and the brain. And we know now that it is vital for homeostasis of both systems or both organs. Germ-free rodent studies indicate that the microbiota is critical for brain development, emotional regulation, and stress response. So in other words, resiliency in the face of stress. And the microbiome has been shown to be a critical mediator in these effects. So much so that most researchers now call this the gut microbiota axis, or the gut microbiome axis, brain axis, excuse me. And so there are four major pathways, depending on, sort of which research you look at. It's typically classified into the four main metabolic pathways by which the gut and the brain can communicate with each other. So we got the metabolic pathway, neurologic, endocrine, and immune. So we've already talked about the metabolic pathway. In other words, the metabolites that the microbiome they make and how that can affect the host system. So short chain fatty acids, we've already mentioned a couple of times that these are really critical anti-inflammatory factors in the body, among other things. Serotonin, 95% of the body's serotonin is converted from L-tryptophan to serotonin in the gut of an animal. And GABA is also some of the microbes can create GABA. So this is gamma aminobutyric acid. And if that's the major inhibitory neurotransmitter in the body. So medications like Xanax, they affect GABA. So that tells you what a powerful effect these can have. So this is going to be a theme that these factors can act locally. So they can have local anti-inflammatory effects, local effects on the microglia, which are the inflammatory cells of the nervous system, but they can also cross the blood-brain barrier. So they can also act, things that are produced in the gut seem to be able to act in the brain as well. So the neural system is another critical pathway through which that can happen. So the second largest group of neurons in the body, or the most outside of the central nervous system, is the enteric nervous system. So it's often called the second brain. It's so complex. And in many ways, it can act on its own. It doesn't necessarily need input from the central nervous system for many of its functions. But that said, it does interact quite a bit with the central nervous system, particularly the vagus nerve. So this is this large meandering nerve associated with the autonomic nervous system. So that, just as a reminder, we've got the sympathetic nervous system, which is fight or flight, freeze or fidget, you could also say. and also the parasympathetic, which is our rest and digest. So the vagus is heavily involved in that. And just an example, this is very complex, but just to pull a couple of examples, the sensory nervous, sensory cells in the gut, they can affect nerve activation in the enteric nerve activation. And the microbiome metabolites can also regulate that neuronal activity of both locally in the enteric nervous system and the vagus, that communication between the brain and the gut. The endocrine system is also very important. So endocrine system largely involves hormones, and that can be many things, but cortisol, so the HPA system, that stands for hypothalamic pituitary adrenal. So cortisol comes from the adrenal glands. So neuropeptides that are created by the microbiome, they can, again, directly stimulate cells within the gut, but they can also modify vagal activity. And then finally, the immune system. We can have the immune system is largely responsible for inflammation, just one example. And we can have local inflammation, but those inflammatory cytokines can also cross the blood-brain barrier and create pathology that way, and also alter vagal activity. So again, just to kind of summarize those pathways, they can have direct effect locally on the tissues and those cells, but they also affect each other. So we can have the metabolites affect neuron firing, and neuron firing can affect the hormones and vice versa. So again, it's a very complex pathway, as in as are most things in the body, it seems like. And so we're just kind of scratching the surface on all that. But it's very fascinating and very promising. So let's talk a little bit more about sort of some negative things like stressors and how that can affect the gut microbiota brain axis. So when the animal experiences stress, there can be a negative cascade effect on the gut microbiota and systems, including the brain. So the stress response, so when the body perceives that there's a stressor in the environment, danger, psychological stressors, there are several things that happen. So we got release of the catecholamines like norepinephrine and epinephrine, but as well as the glucocorticoids that we just discussed. And these can do things like create inflammation and that leaky gut. And that can allow bacteria to translocate. That's just one example. But can also change the gut environment so that it makes it more favorable for the pathogenic bacteria to survive and less favorable for those beneficial bacteria. So that can create dysbiosis, and therefore you have fewer of those beneficial metabolites like the anti-inflammatory short-chain fatty acids and GABA. So that in turn, when you don't have that regulation of the immune system and inflammation, and in fact, you might have pro-inflammatory cytokines affecting or crossing the blood-brain barrier, we can have neuroinflammation, and that can create pathology. So there have been links between, there have been correlates to disease and that pathway. So dysbiosis through some of those mechanisms I just discussed have been linked to things like schizophrenia, Alzheimer's disease, Parkinson's, even autism spectrum disorder. And interestingly, many of these diseases do have a GI disease as a common comorbidity. So many mice studies and rodent model studies have shown that you can take the microbiota of and the contents from an animal such as a mouse that has been genetically engineered to show signs and other histologic markers of Alzheimer's. You can simply take the gut microbiome from those animals, transplant them into healthy mice, and recapitulate that disease or induce that disease by doing nothing else other than transplanting that microbiota. So those studies were first started in sort of the late 90s and early 2000s. And that sort of set off a whole interesting, that's how people started to study this and realize that the gut microbiome is actually tied to a lot of diseases. But sort of zooming back down into what could this mean for our companion animals? Have there been any studies outside of rodent models? So there have been a few studies looking at dogs in particular, attempting to find some correlation between emotionally and behaviorally healthy dogs and those that seem to be unstable or have some issues. And all of these studies, like here's one in shelter dogs that show aggression to other dogs in the shelter. Dogs, this one was a little bit more of a sort of a wide range, just carpet bombing and asking lots of different types of issues. I'm looking at both aggressive and phobic dogs, just sort of a wide lens, if you will. And then another one looking at the microbiome and working dogs, and those that are successful and those that show resilience and those that don't. So all of these studies showed that there was a difference. So within these populations, there was a statistically significant difference in the microbiome between the healthy and the unhealthy animals, behaviorally in this case. Unfortunately, there wasn't a good explanation. So it was really hard to kind of retrofit what they found and explain that with what we know already about the microbiome or the microbiota that were different between those populations. And in fact, something interesting that kind of popped up is in the animals that are unhealthy, so emotionally dysregulated, oftentimes they had higher levels of lactobacillus, which has been shown to secrete GABA, be important in that GABA pathway. So that was a little confusing. More studies definitely need to be done. And this is, sort of behavior is complicated. We know that there's lots of environmental and genetic influences into that trait. So I think we, as the specialists, need to work even further to make sure that we are classifying or phenotyping these animals correctly as well. So it's definitely beyond the scope of this talk to talk about all the interesting research surrounding other metabolic and organ pathologies that have been associated with dysbiosis. There are a few of the diseases or states that are listed here that did have some significant differences in the microbiome in cats and or dogs. So breed was shown to have different microbiota, statistically significant, epilepsy, obesity, metabolic syndrome such as diabetes 2, and then organopathies, chronic kidney disease, liver disease, certain cardiac dysfunction, and then also interestingly, response to treatment seemed to be, that's a trait that may have, how they respond might depend on their microbiome. So that's all really fascinating. And there's one study out of Germany, I believe, where they actually, speaking of response to treatment, when they took this is a very small pilot study, but when these researchers took the microbiome of the dogs that responded to treatment for their epilepsy and transplanted it into the gut of the dogs that were not responding very well, it had a significant improvement. The result was a significant improvement in their response to treatment. So again, all really cool stuff going on and we'll talk a little bit more about these strategies that maybe we can use to support treatment therapies. So what are some strategies to modulate microbiota? So in addition to the fecal transplant that I've just been discussing, diet, that's one of the oldest and probably one of the best ways to influence our microbiome. Probiotics and prebiotics, and we'll go over those definitions. And when you put those together in a product, it's called a symbiotic, and then postbiotics. So let's talk about what I mean when I use those terms. So a probiotic is a living microorganism that, when ingested, conveys a health benefit to the host. So again, how does it do that? It can compete with the pathologic microorganisms in the gut and modulate the immune function and create this healthy barrier. Prebiotics are non-digestible carbohydrates, so basically plant-based fibers. So they're not digestible by the host, but they can be fermented by the microbiota, primarily in the colon. And again, this provides food essentially for the beneficial microbiota, and therefore we're going to have more of their beneficial metabolites. So this is just a picture of a Jerusalem artichoke, because inulin made from that plant is a very common prebiotic, as is psyllium. So symbiotics, again, they are a combination in one capsule, for example, or powder. You've got the prebiotic and the probiotics together. So I've already referred to a little bit to fecal microbiome transplant. So that is defined as transferring fecal material from a healthy donor straight into the intestines of a patient with a specific disease to provide live beneficial bacteria. So this is most effective as it's directly placed into the colon, but some companies are selling capsules that can be swallowed as well. I have not seen as much research on that as the direct fecal microbiome transplant into the colon. So in a clinical setting, this is most commonly used for patients that have dysbiosis and suffering from severe chronic GI disease, such as Crohn's and IBD. Although we have to be careful, because it has been shown that the worse the disease, the worse efficacious, the lower the efficacy, I should say, of this technique. So certainly there are some research into many successes. There's a really interesting clinical study going on with autism. But of course, we want to focus on our companion animals. And there are a couple of studies out there like this one, that puppies that were suffering from parvo, they had an improved outcome after fecal microbiota transplant from healthy dogs. So there are many successes. However, we have to be super careful, because this is not risk-free. In fact, there are reports of human recipients becoming extremely ill and even dying after transfer after these FMTs. And the thought is that they were an unknown antibiotic-resistant microbe was transplanted into this immune-compromised person. So what is a postbiotic? So there's no one definition, but basically postbiotics refer to the administration of the beneficial microbiome products, so the metabolite directly. To be called a postbiotic therapy, it should support host health. And the benefit may be that we are providing the effect of the probiotic without some of these dangers, like I just mentioned, of transplanting live bacteria into a recipient. So the last category, and probably the oldest method right there, is to alter the gut microbiome through the diet directly. So we know from Human studies that fermented foods are an excellent source of probiotics for people. And the last few decades have really elucidated this association between high fat, high saturated fat, I should say, diets and high sugar, low fiber diets, often called the Western diet, because of this and the systemic health risk compared to a Mediterranean diet of complex carbs, in other words, vegetables, and unsaturated fats. So the Western diet is often called a pro-inflammatory diet. And there are lots of studies looking at systemic health effects of these two. I thought I'd pull out just this one little one about brain health and Alzheimer's, and maybe why it's a risk factor for Alzheimer's. So this is just these are MRIs from older patients that are, some have been on a Mediterranean diet and some on a Western diet, and you can see the reduced activity in the patients that had been eating one of those pro-inflammatory Western diets. And if you were to look at, say, PET scans that picked up on the beta amyloid plaques, for example, or other histologic markers of neural inflammation, you would probably see the Western diet had higher levels of those markers compared to the Mediterranean diet. So this has definitely been shown in rodent models and to some extent in dogs. So veterinary nutrition companies have been working on adding these beneficial, anti-inflammatory nutrients. But as the research expands, we've been kind of focusing on the probiotics to complement all of those other types of therapies. So there's some great veterinary examples out there. So Purina has FortiFlora, which contains Enterococcus faecium, the SF68 strain. So this is best known for use in GI issues. So, enteritis and colitis in cats and dogs. But I want to take a back up for a second and take a second to discuss how medications or xenobiotics can affect the microbiome. So no surprise that antibiotics, which kill bacteria, can definitely affect the microbiome. But also remember proton pump inhibitors, even NSAIDs, steroids, of course, all of these can affect the microbiome, potentially in a negative way. So let's focus on metronidazole for a second. So this has been used for decades as the first-line treatment for acute diarrhea in cats and dogs. But what we've seen through some really great research studies is this doesn't just immediately affect the microbiome. It can make what we think are dysbiotic changes for months after the treatment has ended. So this seems to be a factor in, it can contribute to antimicrobial resistance. But the use of through the use of these broad spectrum antibiotics, but they can also actually trigger diarrhea in certain animals. And in fact, we've used metronidazole in studies. So, and the references are at the end of this talk if you would like to read a little bit more about the details of these studies I'm about to briefly talk about. But in dogs that had diarrhea induced by metronidazole use, and cats that had diarrhea induced by amoxicillin clavulanate use, those that were given the FortiFlora product, they have fewer days of diarrhea associated with the antibiotics. Just as an aside, the canine FortiFlora here has been shown to reduce flatulence in dogs. And then in cats, these were given to the Enterococcus, the FortiFlora was given to kittens, some kittens that entered the shelter. And of course, we know that diarrhea is very common with these changes and entrance into the shelters. So kittens that were given the FortiFlora with their food, they had a lower incidence of diarrhea But interestingly, they also measured the mucosal immunity, the mucosal antibody IgA, and that was higher in these kittens. So this led folks to look a little bit more at this involvement of the immune system. So we mentioned already the interaction of the nervous system, the enteric gut system, endocrine and immune systems, and how they can influence each other. So several researchers have conducted studies looking at the effects specifically of these probiotics on the immune system. Like Dr. Lappin at Colorado State found that, giving FortiFlora alone had some enhanced T cell effects in cats. He also has a colony of cats with feline herpes virus 1, and it was shown to decrease the morbidity in those cats when they were given FortiFlora with their meals. Other studies, just briefly, some puppies that were shown to have higher levels of IgA and IgG, specific, I believe, to the distemper vaccine. And then also B cell priming in puppies given that distemper vaccine. And then an older study using what was then called VSL number 3. This was a combination of strains, and it was given to dogs with IBD. And when given this product, they had improved histologic and immune markers compared to dogs given prednisone and metronidazole combination for their IBD signs. So what about the symbiotics? So Purina has FortiFlora Pro, which is the same Enterococcus faecium SF68, but it also includes a prebiotic fiber. And my clinical impressions are it's going to have the same effects of the FortiFlora, but it's just going to be enhanced by providing that fiber. And it has been shown to improve the fecal score in dogs and cats. There was a study with cats as well. And then another product out there that has been shown to have similar effects, another symbiotic product, Nutramax has FortiFlora, and this is 7 strains of bacteria with the prebiotics as well. So dogs that were given metronidazole and enrofloxacin, they had a better long-term appetite and fecal score compared to when with the antibiotic alone. And cats that were given clindamycin had less vomiting and better appetite compared to antibiotic alone. So we talked a little bit about the gut microbiota brain axis. So do we have anything that can be, any of these probiotics that can be considered psychobiotics? And the answer is many have been shown to have some promising effects. One of them, Bifidobacterium longum, has been one of its metabolites enhances the formation of GABA. It is mediated through the vagus, and rodent studies demonstrated that if the vagus was not intact, then its effects were not as strong. And in these studies with both rodents and in people that were administered several weeks of Bifidobacterium longum, they had a lower response when exposed to stressors compared to when they did not have that Bifidobacterium longum administered. So Purina has Calming Care, which has a specific strain of Bifidobacterium longum called BL999. And they have some nice controlled studies to show that when their research dogs were administered, and these were blinded studies, when they were administered the Calming Care, they had reduced fear reactions to many different types of kind of fear-inducing stimuli, such as noise stressors, stranger approach, separated from their caretakers compared to when they were not given that product with their diet. And Purina Calming Care is also available for cats. So the same Bifidobacterium longum 999, again in a study with Dr. Lappin's lab and his feline herpes virus infected cats, when they were administered calming care and after they went through a stressor compared to when they were not given the calming care, there were signs they were friendlier when they had the calming care. So the increased contact seeking with their caretakers, less pacing in their cages on calming care, less of an increase in cortisol, and then lower morbidity associated with their upper respiratory infection while on the calming care. So all very interesting. So there are also some veterinary diets formulated to support the pet's microbiome. So these are typically prescription diets that contain the appropriate amount and ratio of fibers as prebiotic sources. Most commonly out there to support pets that are experiencing large bowel diarrhea. So Purina has EN fiber balance for dogs. Hills has GI biome for cats and dogs, and Royal Canin's has GI fiber response. So all these companies are working hard to find appropriate prebiotic in their maintenance diets as well. So there's so much fascinating research on this topic. And if you're interested in learning more, particularly with respect to cats and dogs, the Purina Institute has an online microbiome forum each year. I believe they have 2 versions of it every year. And you can watch these on demand at no charge. So the speakers cover a wide range of research topics, some related to companion animals, but they're not specific to any product. So other veterinary nutrition companies also have some similar forums in the past as well. So in conclusion, the microbiome is actively involved in host GI and other systemic physiologic functions. Metabolites provide other anti-inflammatory and other regulatory factors. Dysbiosis is associated with many disease states in the host, but we have to be careful about assuming causation. At this point, for many of these, we can say there's a correlation, and only a few do we really know that pathway and causation. So manipulation of the microbiome, though, may be a promising therapy to improve host health, but much more research is needed on the best ways to safely manipulate the microbiome as one type of therapy.